Hello friends, Ace Botane, next episode. In this episode, we'll be discussing first episode for the molecular basis of genetics. That is, second chapter of genetics, more towards biochemical aspect. Again, for students, it will be tougher. So we can proceed without wasting time. So I will share the screen for your convenience. So it will be easy. I think screen okay. So we can proceed. So molecular base of inheritance, one, what all things we'll be discussing in this episode. First, as usual, introduction will be there. Then, second is DNA. Third is questions. So, three topics we'll be discussing. Introduction, DNA, question. Introduction already given here. So, we can proceed. Again, remember this is more towards biochemical aspect, more modern than Mendel. In Mendelism, we already familiar with the term factor. And Mendel's factor is today what we call it as gene. And who will carry the DNA? Chromosome will carry the DNA. And how genes and DNA are linked? Genes are segments of DNA connected with the coding for a specific chemical like proteins, etc. So, chromosome, DNA, genes. I think that idea is clear to all of you. Now we can continue. Previously in the sense that Mendelian time or connected with that time period, no idea about which is the genetic material. And this quest and dilemma made it more and more confusing. So more and more interest developed. And around in 1926, this quest and dilemma for genetic material reached into the molecular level, reaching into prediction and realities that in majority of organism, DNA is the genetic material. That is how that idea came. DNA will be the genetic material. Now, when we say DNA, many other chemicals like RNA, role of RNA, role of DNA, how the duplication takes place in both, specifically DNA, and what is transcription, what is translation, what is gene regulation. And after that, HGP, Human Genome Project, and FP, fingerprinting. This all established gradually, so we are completely becoming modern genetical area. That is how that introduction goes. I think idea clear to all of you. So which all topic we'll be discussing in this chapter, everything. Now DNA. Now DNA, when we discuss three scientists' name, at least we remember. One is G.J. Mendel. See the time where they are living, 1822 to 1884. Charles Darwin, 1809 to 1882. And Frederick Meister, 1844 to 1895. So they have almost contemporary. And DNA was first identified by Frederick Meister in 1869. And who given the term nuclein. And it was acidic in nature. Acidity of this chemical was given by originally Altman. And he also proposed the term nucleic acid instead of nuclein by Frederick Meister. Now, what is DNA? Expansion of DNA, deoxyribonucleic acid. And what is the meaning of it? DE normally means minus, inferior, negative, etc. So meaning is one oxygen less in second carbon atom. That is why this term 2 dash. Atom in comparison with the RNA. Comparing with RNA, one oxygen less in second carbon atom. And what is originally DNA? DNA is nothing but a long polymer of deoxyribonucleotides. Now, what are the main components of DNA? Basically, three components we can find. Nitrogen base, sugar, phosphate. 3. Nitrogen base, first item, here we can find it. Nitrogen base plus sugar, it is called nucleoside. Nitrogen base, sugar plus phosphate, it is called nucleotide. Nitrogenous bases can be 2 in connection with the DNA, purines and pyrimidines. That we'll be learning later. Now we can proceed to next page, hoping that it is clear to all of you. Second page. Thymine. Thymine is a nitrogen base Thymine is originally 5-methyl uracil. This is a repeated question. This one. Thymine is 5-methyl uracil. Now, when NCERT says some data, it is important either as match the following or direct or odd one exception likewise. So first item, bacteria phase X174, where we can find single-stranded DNA, not so common. Shape is circular, and where you can find 5, 3, 8, 6 nucleotides or bases, no pair, single-stranded. Bacteriophage lambda, double-stranded DNA, linear, 48502 base pairs. 48502 base pairs, since it is double-stranded DNA. Now, E. coli. E. coli again double-stranded DNA, circular in shape, 4.6 into 10 raised to 6 base pairs. And finally, human genome, double-stranded DNA, linear, 3.3 into 10 raised to 9 base pairs. So in bacteriophage X174, since it is single-stranded, we can find no base pair rule is applicable. That will be learning later. Bacteriophage X174 is a virus infecting bacteria. Normally in viruses only we can find single-stranded DNA. Genome, what is meant by genome? Here the term human genome. Genome means haploid set of chromosome, that is in a gamete or microbes or in each cell of multicellular organism. Haploid set. That is called genome, haploid set of chromosome. Now comes purines and pyrimidines, their comparison, differences, etc. What are purines? Purine is an hydrogen base where we can find adenine and guanine. It is basically an hydrogen base from purines. It is a double ring structure, 9-membered, nitrogen at position 1379. It is heterocyclic. Now pyrimidines, thymine, cytosine, and uracil. It is single ring structure. Previously, purine, double ring, it is single ring here. 6-membered, previously 9-membered. Nitrogen at position 13. Previously, or purines, 1379, here it is only 13. Heterocyclic. And thymine is again 5-methyl uracil. I think that idea clear. Now we can proceed to main bonds in DNA includes basically 3 main bonds. One is glycosidic linkage. Second is phosphoester or phosphodiester linkage. Third is hydrogen bonds. This is typical NCERT diagram. Diagram-wise question can be asked. Labeling question, where the hydrogen bond, where the glycosidic bond, where the phosphodiester bond, how many hydrogen bonds are available? This way. Phosphate comes at which end? OH comes at which end? 5' end or 3' end? This way it can be asked. NCERT diagram. Now we can proceed to next page, where same diagram I given some numbers, etc. So first is hydrogen bonds. Where we can find this hydrogen bond? The color, if you notice, we can find brown color, I think. Here we can find hydrogen bonds connecting two nitrogen bases. Then phosphoester or phosphodiester. What is the purpose of that bond? Connecting phosphate to nucleoside through sugar. So color is red or orange. So we can find here it is marked. Phosphoester or diester. This one. P. Here. Okay. Now, phosphodiester bonds connecting phosphate to nucleoside through sugar and thus form nucleotide. Hydrogen base plus sugar, nucleoside. Nucleoside plus phosphate, nucleotide. Through phosphoester linkage of alcohol of 5 carbon, 5th carbon. 2 nucleotides are linked through this. Another important question, NCERT 3' to 5' phosphodiester linkage. So here, diester linkage. And that forms a dinucleotide. Now comes third bond, N-glycosidic linkage. That is connecting nitrogen base to sugar. Nitrogen base, adenine, guanine, etc. to sugar. So this one here, that bond comes. Adenine, guanine, thymine, etc. nitrogen base to the sugar. That is N-glycosidic linkage connecting nitrogen base to sugar to form nucleoside. Color we can find. Linkage is through OH of 1st carbon of pentose sugar through this linkage, purine nucleosides have 1' to 9 means that attachment direction 1' to 9, that means carbon first of the sugar and the 9th position of adenine or guanine. While in pyrimidine, nucleoside have carbon first of sugar to first position of thymine or cytosine, that is 1' to 1. I think I did clear to all of you. Okay, now we can proceed to a diagrammatic representation of it. So it will be clear. See, this is sugar, this is nitrogen base, linkage you can find here, hydrogen bond you can find, and here comes phosphodiester bond connecting with the phosphoric acid. Phosphoric acid is H3PO4. So P only one, 3H, 4O. And it will be linked with the OH of sugar. And we can find here one water molecule will be eliminated, here one water molecule will be eliminated, so two water molecules will be eliminated, and that forms a diester bond. And that is how this is the bond formation. And that is how they are linked, phosphodiester bond, nucleotide formation. And ribose is the molecular formula is C5H10O5. That is an RNA ribose. Deoxyribose in DNA, deoxy, one oxygen less. That is why C5H10O4. I think it is clear to all of you. Now comes RNA DNA, adenine base sugar and phosphate. Here, adenine, guanine, thymine, cytosine, adenine, guanine, cytosine, and uracil. Thymine will not be there in RNA. When adenine base couples with the sugar, it is called adenosine, guanosine, cytidine, uridine. And it combines with the phosphate form adenylic acid, guanylic acid, cyclidic acid, uridylic acid. So this way question will be there normally asking adenosine, which is an hydrogen base. Likewise. Now coming to DNA. DNA, adenine, sugar is deoxy. When these two couples, deoxyadenosine, deoxyadenylic acid. Guanine, deoxyguanosine, deoxyguanylic acid, thymine, deoxythymidine, deoxythymidilic acid, cytosine, deoxycytidine, deoxycytilic acid. A question they are normally asking. Now we can proceed to next one. Types of DNA. When we come to types of DNA, another usual question. Scientists say more than a dozen DNAs will be available. And for us, basically, 5A, B, C, D, E, Z, 6. 6 will be there. Everything I written here should be easy. See, all these are right-handed except Z. It is left-handed. And number of bases per turn. A, 11 base pair. B, that is Watson DNA, 10 base pair. C, 9 base pair. D, 8 base pair. E, 7 base pair. And Z is 12 base pair. And it is left-handed. And here remember, some decimals will be also there for each base pair. In some cases, decimals will be there. Now comes derivation of DNA structure. How this has been derived. Different lines of studies were parallelly conducted by different scientists of the world. And finally, two lines of studies were emerging. One was X-ray crystallography, and second is famous Charkov's rule. So first is X-ray crystallography. And it is basically done by a scientist, Maurice Wilkins, along with Maurice Wilkins, second scientist, Rosalind Franklin. Diffraction studies, a sort of helix with a 3.4 Armstrong periodicity, they identified, but not suggested a definite model. Now comes a famous rule. We cannot say rule, still. Erwin Charkov's rule, or Charkov's rule, only applicable to double-stranded DNA. So it is basically some speculations, or more specifically some generalization. It is applicable to only double-stranded DNA. That's an important point. And what are the main connotations? Purines equals pyrimidines. Purines in DNA includes adenine and guanine. Pyrimidine in DNA includes thymine, cytosine. Purine, pyrimidine are equal in amounts. That is why adenine plus guanine. Purine equals thymine plus cytosine. Or we can say A plus G by T plus C equals 1. And this value will be constant for all species in the world. Constant, specifically constant. So value 1 and constant, that is different. A plus G by T plus C. We can find it is 1. While A plus T by C plus G. So once again, purines equals pyrimidine. A plus G equals T plus C. So A plus G divided by T plus C equals 1. And this is constant for all species. Second, A plus T divided by C plus G. So this value and these combinations are different. Very specific for a species. It may not be 1. It is specific. And this specific value can be used for identification. And in prokaryote, this value will be low, less than 1. So we can find advancement. In eukaryote, this value will be comparatively higher. Typical example in humans, it is 1.52, while in Escherichia coli, it is 0.92. Based on these principles, we can find phosphates, deoxyribose sugar are equal in number. And these are the two famous scientists that we'll be studying later, Watson and Crick. Watson, Crick. Nobel Prize received in 1962. They connected with the B-type DNA, most famous DNA. Now, next aspect is some historical event and connected with it how things came. In 1953, James Watson, Francis Crick, based on X-ray diffraction data produced by Maurice Wilkins and Rosalind. Previously, we discussed the term, scientist, Rosalind Franklin, proposed a very simple but famous double helical model of DNA, this one. For the structure of DNA, NCERT, page number 81, 2024, March 31st, reprinted edition, or March reprinted edition. 1. DNA double helix is anti-parallel. So what is meant by anti-parallel? See, here you can find. Here you can find. This is supposed to be anti-parallel. See, anti-parallel. One is 3' to 5', other strand is 5' to 3'. One strand is 5' to 3', while other is 3' to 5'. 2. Backbone. Backbone means this one. It is made up of sugar and phosphate. Rungs. Rungs are steps. They are made up of nitrogen bases with the hydrogen bonds. Purine always comes opposite to pyrimidines. It is for maintaining equal distance between the two strands of DNA. Bases will project inside. See, it is projecting inside. These are bases projecting inside. A with the T, 2 hydrogen bond. G with the C, 3 hydrogen bond. GC, AT. 5. DNA DNA helix is right-handed. BDNA diameter is 20 Armstrong, radius is 10 Armstrong. Pitch of the helix is 3.4 nanometer turns. And a nanometer is 1 billionth of a meter. Another common question, 10 raised to minus 9 meter. In BDNA, each turn of double helix has 10 pairs. Distance between two base pairs will be either 3.4 Armstrong or 0.34 nanometer. If we take 3.4 Armstrong, 10 base pairs means 34 Armstrong. Suppose if we say nanometer, 0.34 into 10, that is 3.4 nanometer for a DNA double helix with the 10 base pairs. I think idea clear. Now, next page. The plane of one base pair stalks over other. In double helix, it will confirm the stability of the DNA. Stacking of DNA is, in the diagram we can observe, stacking one above the other. Now, one of the hallmarks of their proposition was base pairing, NCERT, between two strands of polynucleotide chain. Again, that is given in NCERT page number 81, March 24, reprinted ish. Now, as usual, questions. You can find 5 theory, 5 NEET model with the key. But here one item I mentioned. See, there are two terms, major grooves and minor grooves. Major grooves means here backbones are far apart. Here you can find distance is small. Backbones are far apart. While minor grooves, see here, backbones are close together. That is how major grooves and minor grooves comes. I think idea clear. So that is regarding the topics for us. So what all things we learned in this episode. Basically, the chapter deals with what items? More biochemical aspect, etc. Then these are the NCERT idea concepts. And what we'll be discussing here is basically introduction, DNA and questions. DNA is a very big area. Introduction, how the theoretical aspect, how it came up to historically, came to fingerprinting, then DNA, expansion, then meaning here 3 scientists, Mendel, Darwin, as well as Meissner, and the Meissner 1869, nuclein, acidic, Altman, nucleic acid, acidicity. Now what is DNA, full form, meaning? Then we entered into main components of DNA, nitrogen base, sugar, phosphate. Now, meanwhile, in between topic, NCERT, organism, genetic material, number of nucleotides or base pair, a common question. Then we entered into bacteriophage, YX174 single standard, no base pair rule is applicable. Then genome, what is the meaning, haploid of chromosome, then purines, pyrimidines, their details, then famous NCERT diagram, 3 bonds, hydrogen bond, glycosidic bond, phosphodiester bond, and where that bonds lies, that aspect, and what will be formed. After that, a typical representation, how this phosphodiester bond comes, how many molecules of what will be eliminated. Then ribose, deoxyribose, a molecular formula, why it is deoxy, here O4, here it is O5. Then nitrogen-based sugar and phosphate comparison, RNA, DNA. Typical example, cytosine, cytidine, citrullic acid, thymine, deoxythymidine, deoxythymidinic acid, that way. Then came types of DNA. Z is exceptional, left-hander, 12 base pair per turn. Restaurant, right-hander, and number of base pairs. Derivation of DNA structure, X-ray crystallography, Charkov's rule. Then Charkov's rule, under which so many items we proceeded. Purine pyrimidine equals same value. Purine DNA includes AG. Pyrimidine includes TC. Purine pyrimidine equivalent amount A plus G equals T plus C, or A plus G by T plus C equals 1. This value will be constant for all species. Then second is A plus T by C plus G is very specific for a species. For identification, we can use it. And that will be value will be higher in eukaryote, lower in prokaryote. Then James Watson and Francis Crick, their contribution. And what are the important items? Antiparallel nature of 2 strands. Backbone is made up of purine, always comes to opposite to pyrimidine, so the distance can be maintained, equal distance between these 2 strands can be maintained. A with a T, 2 hydrogen bond, G with a C, 3 hydrogen bond. And B DNA is a typical DNA, it is right-handed. B DNA diameter 20 Armstrong, radius 10 Armstrong. Pitch of the helix is 3.4 nanometer. A nanometer is 1 billionth of a meter, 10 raised to minus 9 meter. Then in bDNA, each turn of double helix has 10 base pairs. So distance between 2 base pairs will be 3.4 Armstrong, or total will be, double helix will be 34 Armstrong. Then plane of 1 base pair stalks over the other. In double helix, it will confirm the stability of the DNA. Stability of the DNA. Then one of the hallmark of their proposition was base pairing between 2 strands of polynuclear chain, NCERT. Then here I mentioned major groove and minor groove. After that, 5 theory question, 5 NEET model question with the key. Hoping that it is clear. If you like this episode, subscribe all, like it, share it, comment on it. We can meet in the next episode. So take care. Bye. Thanks.